So it's my pleasure to introduce Dr. Tina Rogers. So she's a life science executive with 25 years experience in early drug development in the CRO industry, serving the biopharma and animal health industries and the NIH and DoD. She's board certified toxicologist with a broad technical background, including cell biology, immunology, toxicology, cell and gene therapy, sepsis, inflammation, BL3 and selective agents, flow cytometry, and predictive in vitro toxicology. In management roles, she has been responsible for several organization projects that drove growth and profitability and created business development organization and served as an advisor to a joint venture, CRO venture in China, and championed several acquisition processes, including 2 EU pharmaceutical companies. She's established new service lines with state-of-the-art technologies for the analysis of protein and genomic biomarkers to support preclinical drug safety and efficacy studies. She serves on several boards, including not-for-profit, academic, and biotech organizations. She is fortunate to have cultivated a broad network of colleagues and clients within the biopharmaceutical and CRO industries in the U.S., Europe, and China. So it's my pleasure to welcome Dr. Tina Rogers. Thank you.
Thank you so much, Kenneth. Can you see my screen?
Yes, we can.
Fantastic. Okay, and thank you also to the organizers at Precision Global for the opportunity to talk to you today. So yesterday at the panel discussion and also just at this meeting in general, we talk about strategies to improve the success of drug discovery to take us ultimately to the clinic. And so today I'm going to talk about some things that you should consider when you're finally ready to do your pivotal IND studies. So just an outline for the presentation today. So we're going to talk about considerations in transitioning from discovery to development, talk about some different kinds of molecules and how those molecule types will dictate some aspects of your IND program. In addition to just large and small molecules, we're going to look at a few other therapeutic classes, and then I will finish with some final recommendations. So if you or your company is new to the world of preclinical development, which is a regulated aspect of drug development for sure, there's some things that I think is important to keep in mind. First of all, the flexibility that you have in your efficacy and exploratory studies doesn't apply to your pivotal IND studies. So what do I mean by that? Your IND studies are driven by a protocol that includes every aspect of what's going to happen in the study and when. So it's very difficult to make changes on the fly in pivotal IND studies. And actually, it's not recommended just because it increases the risk to your program. Also, keep in mind that any GLP studies that you run are going to have to be included in your regulatory submissions. So if you start a study and your animals start dying or your study wasn't well designed, you can't just stop it and start over. So that's a good thing to keep in mind. Also, so if you have just in-licensed an asset, or if you're moving to a different CRO, or you're wanting to start your GLP studies, it's always a good idea to start with a non-GLP study if the existing data that you have aren't recent and aren't from a lab that you're familiar with. It's just never worth the risk to jump right into a GLP study. Also, if your company doesn't have the regulatory consultants that you're going to need for your specific type of molecule and indication, there are lots of great regulatory consultants out there that they generally specialize in certain areas. So just be sure that you're choosing someone who can really help you with your specific situation. Also, sort of related to the flexibility bullet at the top, it's never a good idea to add exploratory or unnecessary endpoints on your GLP studies, because you may end up with more questions than answers. And so GLP studies are not the time to explore. again, avoid non-GLP contributions on your GLP reports. They're just difficult in terms of expectations that the regulators have. And if you have to use non-GLP contributions, just make them as GLP-like as possible and communicate with that lab, if it happens to be an academic lab, what the expectations are. And always take advantage of your opportunity to meet with the FDA for your pre-IND meeting. It's a great opportunity and your only opportunity to get feedback early on and make sure that you address all the primary areas of CMC, preclinical, and clinical. So just starting with the major different kinds of molecules, small versus large molecules, and how, at least in the context of drug development, what are their differences? So small molecules are obviously chemicals. They're chemically synthesized. They're low molecular weight. They're active in many species. They're generally not immunogenic. They undergo metabolism by the phase 1 and 2 drug metabolizing enzymes. And their toxicity is either from the parent drug or from a metabolite. Whereas biopharmaceuticals or large molecules are, by definition, they're produced in living cells. They have a high molecular weight. They're target specific. So that means that your safety evaluation, that is your tox studies, need to be done in pharmacologically relevant species. Biopharmaceuticals are potentially immunogenic, so you need to address that as by doing ADA or anti-drug antibody assays in your tox programs. Biopharmaceuticals undergo proteolytic digestion as opposed to metabolism like the chemical drugs do. And toxicity of biopharmaceuticals is due generally to their exaggerated pharmacology. So because of the difference in toxicity of these two classes of molecules, as well as their differences in terms of targeting, being specific about targets, there are differences in the requirements for your IND program. So small molecules need to include species that are relevant in terms of metabolism. And generally, both rodents and non-rodents are required for a small molecule program. Your dose selection is based on maximum tolerated dose, as well as your intended clinical dose. And pivotal toxicology duration is from 2 to 4 weeks. And then safety pharmacology, which tests the effects of your drug on cardiovascular, respiratory, and CNS systems. These are usually require standalone studies for small molecules. And genetic toxicology studies, or gene tox, is required. So for large molecules, the species is selected based on pharmacology. It needs to be a relevant species. And if there's only one relevant species, you may only need to run one species in your program. Your dose selection is based on the maximum pharmacological effect. As I said earlier, one species might be sufficient, and generally the pivotal IND studies for large molecules are 4 weeks. And generally, standalone safety pharmacology studies and gene tox are not required. For safety pharm, the endpoints can be incorporated into your tox studies. So this is a gantt chart, showing some of the studies that need to be done for small molecule IND programs. And this obviously doesn't include all of the CMC information that Paul O'Shea talked about yesterday. But this program runs for about 26 weeks, about 6 months. And the dark bars at the top represent the method development and validation of your dose formulation, as well as the bioanalytical method developments for 2 species, rat and dog. And the lighter bars at the bottom represent the animal studies, the individual animal studies that need to be done. You'll do some initial dose MTD and 7-day repeat dose studies in your 2 species, and your pivotal studies are your 28-day repeat dose tox studies in this case. You can get away with 14-day in some circumstances, depending on what your clinical plan is. And then here at the bottom, you have the central nervous system and cardiovascular and respiratory safety pharmacology studies that need to be done. So an important takeaway from this is that notice that you really need to start your chemistry in advance, maybe a month in advance of your in vivo studies. And so this is a similar chart showing a large molecule IND program for like a typical monoclonal antibody. And in this case, we're showing that 2 species are run, but as I said before, you may not need to run 2 species. So the first thing you notice compared to the previous slide is how many more method developments and validations have to be done for large molecules. And in addition to that, these are generally ligand binding assays, LBAs, that are required for these methods, and they're more complicated, and sometimes they're more difficult to develop and validate. So you really need to start really further in advance with these, even than the small molecule drugs. The advantage here is that you do only have to run either 2 or 4 in vivo studies, as opposed to the more studies that were shown on the previous slide. But you will also notice that overall this program runs for about 9 months as opposed to 6 months, and that's really just because of all the upfront chemistry that's required. So these, now moving on to some other therapeutic classes. So let's start with oligonucleotides. And oligos sort of fall in between small molecules and large molecules. They're chemically synthesized like small molecules, but they can also be target and species specific like large molecules. And oligonucleotides can be either double-stranded or single-stranded, as shown here. And there are lots of different kinds of oligos. Most of the ones that are clinically being pursued either promote RNA degradation here, or they inhibit translation. The CpG oligos are a different sort of animal in that they activate innate immunity through the toll-like receptor 9, and they're used as adjuncts or as adjuvants for other therapeutics. So in terms of toxicities for oligonucleotides, toxicities are either hybridization dependent or hybridization independent. And for the hybridization dependent toxicities, it's usually an exaggerated pharmacodynamic effect, which is obviously target and species specific. Now, oligos can have some off-target hybridization in that they're hybridizing to a sequence that you don't intend them to. And these can generally be identified through in silico and in vitro methods. So most of the toxicities coming from oligonucleotides are hybridization independent, and they can either be sequence dependent, like thrombocytopenia, or sequence independent, like the effects on coag and complement, and they can fall somewhere in between, like the effects of immunostimulation and effects of oligos on high exposure organs like the liver and the kidney. These effects can be predicted generally by in vitro assays with human plasma or cells, or in long-term non-human primate studies. And most of these hybridization-independent toxicities are generally due to protein binding. So next we'll move on to bispecific antibodies. We heard some of this yesterday from Dr. Jagath. So bispecifics, just like it sounds, they're engineered to bind to 2 different antigens at the same time, and that improves their specificity. So there are many designs of these molecules, but they fall into 2 different basic categories. They're IgG-like, which are stable molecules with a long half-life, but their size limits their tissue distribution. The other main class is the fragments that have no FC region, and bites are an example of these. Fragments are less stable, so they have a shorter half-life. That means they require frequent dosing or infusions, but their small size leads to an increased tissue penetration, so that's really an advantage. And there are 3 FDA-approved bispecifics, and this recent one, Rybrevant, I think was just actually approved last month for lung cancer. So bispecifics can result in some development challenges. Their toxicity is unpredictable, and it's usually related to an immune response. Can cause symptoms that are listed here, including anaphylaxis, cytokine release, and mortality at high doses. The toxicity can be mitigated using either multiple ascending doses, loading doses of the bispecific before the larger dose, and also anti-inflammatory steroids and antihistamines. So an additional development challenge for these is that they're highly immunogenic, more immunogenic than monospecifics. So they may impact your PK at all dose levels instead of just the low dose, as with many monospecifics. Again, loading doses of the bispecific can reduce immunogenicity. They're very potent molecules, so they require really low doses. This means that they're probably also going to require slow bolus, or more often than that, infusions for administration. So this presents a challenge for bioanalysis because the low doses result in very low plasma levels, so that ELISA and even the electrochemiluminescent methods are sometimes not sensitive enough to pick them up. But there are more ultra-sensitive technologies, and these technologies are getting better over time, because this is a really it's a fast-growing area. So next, antibody-drug conjugates. We heard about those yesterday as well. This is a really fast-growing category of anti-cancer drugs. So as the name implies, these are conjugates of high-affinity binding antibodies with a payload drug attached to them using a linker. So the payloads are drugs that either disrupt microtubules or they damage DNA, and some of them are shown down here. So the whole point of antibody-drug conjugates is it reduces the systemic exposure to these cytotoxic payloads. So if that is going to be true, then your linker is very important, and the stability of that linker is important. They can either be cleavable or non-cleavable, and you want them to cleave in the right place once they reach their target, not just out in the plasma. These are very potent molecules, and their toxicity is unpredictable as well. So for regulatory issues here, there's generally no need for studies with the monoclonal antibody or the linker alone. However, the payload needs to be very well characterized in terms of toxicity, and often these payloads are reused, so they're already marketed, and so you don't have to repeat that. But if it's a new payload, or there may be other reasons why you're going to need to include, either run a whole program for the payload or just add an arm onto your study with your ADC. So as I said before about the linker, the stability of that linker is really important. So in vitro plasma stability is needed for an IND for these. So yesterday, Dr. Jagat said there are 10 approved ADCs, so I must have missed one. And there are many more in clinical trials. So my final category is gene and cell therapies, and I've combined these just because they have a lot of similarities in terms of the different approach. So both of these therapies, they don't employ standard testing approaches because they often can't. So often non-standard animals or even diseased models can be used for toxicology studies. And if there's only one single species that's relevant, then only a single species can be used, is sufficient. One thing to point out is that the potency of these is not only about concentration, in other words, the viral genomes or cells per mil, But these are activity-based, so you need to make sure for your gene therapies that they do indeed cause transduction, and it needs to be defined in terms of transduction efficiency. And for your cells, for cell therapy, they're not just cells per mil, but they need to be living cells per mil. These are just things that don't occur for small molecules and normal large molecules. Again, safety pharm and gene tox are not routinely required. Stem cell therapies will require tumorigenicity studies just to make sure that there's no uncontrolled growth in the body after administration. So also for cell therapy related to these are living cells, and you need to make sure that your cells are going to survive the trip from the manufacturer to your testing lab. It's always good to do a test shipment. Often these are for rare disease indications, and so infant and juvenile NHPs may be required, and that's going to affect your lead time. If your gene therapy is an AAV vector, you're going to need to pre-screen your animals for neutralizing antibodies, and you're going to expect more than 50% positive, meaning that you can use less than 50% of the animals. So you just need to keep that in mind and test a large pool. And if you're doing CRISPR studies, you also have to pre-screen the animals for your SNPs that are needed for editing. So schedule the AAV neutralizing antibody screening in advance. That can be a bottleneck. For gene therapy studies, immunogenicity assay development can be complicated because you're going to have to look at anti-vector and anti-transgene product antibodies. You may need surgery expertise for biopsies for these studies. And whatever lab is doing your tox studies need to be able to do aseptic and efficient necropsies to avoid contamination of the tissues and from tissue to tissue and animal to animal, and to preserve the integrity of the therapeutic. And as opposed to the LCMS and ELISA for like bioanalysis of large molecules and small molecules, the biodistribution is done with PCR in tissues for cell and gene therapies. Okay, so my final slide is just recommendations as you move towards your pivotal IND studies. Plan ahead. This has been mentioned already yesterday by Paul O'Shea. Your manufacturing, your method development, and even acquisition of your animal model, if it happens to be an NHP, can affect your starting date. There's no substitute for experience. Make sure that you hire or have relevant regulatory experience, especially if you're in a niche area. Always share the data that you have with your preclinical team or your CRO, because the more they know about the drug, the better they can design the studies. And again, always start with non-GLP studies if you're moving into a new species, or if it's an in-licensed asset, or if the previous studies have just been done too long ago or at a lab that maybe doesn't even exist anymore. Don't try to save money by decreasing animal numbers or dose groups in your tox studies. It just increases risk, and it can cause a lot of heartburn, and you may end up repeating the study anyway. And keep your exploratory endpoints in non-GLP studies, because they may provide more questions than answers for you. So bottom line here is that GLP studies are a commitment, and be prepared and know what's realistic before you start them. So that's all I have today. Thank you very much, and I'm not sure if we have time for questions.
Okay, well, yes, and thanks so much for your talk. I think we have a little bit of time. The next talk is until 10:35, so if anybody has any questions, we'd like to answer. If not, can move on to the next, or I can ask a quick question. So you mentioned in the gene therapy, the response to the efficacy was more due to not the concentration, but the kind of viral load. How is that typically determined? Do you just kind of spread it out and see how what the response is to the gene therapy, and then use that to design the GLP trials? Or
So I guess my point was, you know, so I work in a CRO, right? And we receive the certificate, the C of A, from the laboratory. And generally, you know, when you're talking about, so you need to understand what concentration you're working with, right, to define your dose levels. And so it's not just about how many viral genomes are there, but are the viral genomes actually doing what they're supposed to do? Right? Are they active? Was your vector constructed the right way? I've actually, you know, done a study in the past where it was a primate study, a gene therapy study, and we realized after the end of the study that the transgene was inserted in the opposite direction, so there was nothing going on. So that was like a totally wasted study. So the point is, you have to really, you know,
Really
these
dig
things are
down, kind of.
complicated, right?
Yeah, that makes sense. Okay. All right. Thanks so much, Sam. I don't know if we move over now.
Okay, okay, thank you.
Yeah, thanks for a great talk.
Thank you, Dr. Tino, for your talk. And yeah, we will be transferring over to the next session now with Dr. Yuxin Wang.